Good morning, sir, and good morning to everyone. I am K. Rupika from the first BTech of the CSC department. Now, I am going to give a seminar on sensors topic. In this video, we are going to discuss about the introduction on sensors, what are the types of sensors, potentiometric sensors, amperometric sensors, their examples, and the conclusion. First of all, what is meant by sensor? Sensor, it is a device which can convert the chemical information present in the sample directly into the electrical signal. Sensor, it is a device which converts the chemical information directly into the electrical signal. Sensors, it provides the information in the form of an assay. Assay means analytical method. It transforms chemical information to electrical signal. There are four types of sensors mainly. They are optical sensors, thermistor sensors, biosensors, and the electrochemical sensors. In these electrochemical sensors, there are three types again divided. The first one is the potentiometric sensor, the second one is the amperometric sensor, and the third one is the conductometric sensor. In this video, I am going to discuss about the potentiometric sensors and the amperometric sensors only with their examples and the conclusion. First one, the potentiometric sensor. It is used to determine the electrode potential or voltage present between two electrodes. These potentiometric sensors are used to determine the electrode potential or the voltage present between two electrodes. In these potentiometric sensors, we observe three electrodes and the electrolytic solution. The three electrodes are the working electrode, the reference electrode, and the counter electrode. Here this is the diagram of the potentiometric sensor diagram. Construction and working process of the potentiometric sensor. We observe here in the diagram, this is the reference electrode and this is the working electrode. Electrolyte present here, and these here we observe the analyte solution and the pH meter. The reference electrode and working electrode are connected to the pH meter. When voltage is applied, the ions are formed from the electrolytic solution and H plus ions are detected by the glass electrode. The difference between the concentration is formed, which is called as EMF. EMF means electromotive force, and this EMF is detected by the pH meter. We can say that the glass electrode is the mother of all electrodes. Here we see the diagram of the glass electrode. Glass electrode diagram is present here. Glass electrode detects the H plus ions, mainly H plus ions in the electrolytic solution. It is due to in the glass electrode there is a presence of glass membrane which creates the difference in the concentration. So this glass electrode mainly detects the H plus ions. The applications of the potentiometric sensors. The potentiometric sensors are used in the gas sensors to detect the toxic gases like ammonia, NH3, NO2 and CO. It is also used to identify impurities present in the water. It is used as ion selective electrodes and it is used as a biosensors. Transduction principle. Transducer. Here transducer is a device which converts the chemical information into the electrical signal. The analyte, it is a substance which used to be detected. Analyte is a substance which used to be detected by the transduction principle. Analysis of a glucose is the example of these potentiometric sensors. Here we observe the glucometer. In this diagram, the glucometer is observed. It is used to detect the glucose from the blood sample. See here, glucometer, device used to detect the glucose from the blood sample. Glucose plus O2 plus H2O gives rise to glucanoic acid plus H2O2. Here H2O2 means hydrogen peroxide. Again these hydrogen peroxide on oxidation process undergoes to give 2H plus O2 plus 2 electrons. The second one of these is amperometric sensors. The construction and working process of the amperometric sensors are, we observe the cell here amperometric cell here. This is the ammeter, and the cathode, anode, and the electrolytic solution is present here. The two electrodes, cathode and anode, are connected to the ammeter in the amperometric cell. Both electrodes are connected to connected to ammeter. In these amperometric sensors, the redox chemical reactions occurs due to the transfer of charge takes place. Redox chemical reaction means that where a reaction undergoes both oxidation and reduction process. Oxidation means loss of electrons, and reduction means gain of electrons. Where both these reduction and oxidation process occur simultaneously in a reaction, this is called a redox chemical reaction. Here it is a reversible chemical reaction. Forward and backward reactions are occurring fastly, and slow resistance is occurring in the reversible chemical reaction. And in reversible chemical reaction, forward reaction is slowly and the backward reaction is fastly. High resistance is observed. When voltage is applied, one part is undergoing oxidation process and other part is undergoing reduction process. So we call it is an redox chemical reaction. Flow of electrodes from glass electrode to reference electrode, that means anode to cathode. Flow of current from cathode to anode, vice versa. These, and when the voltage is applied, electric current is generated. This electric current is detected by the ammeter as shown in the figure. Here the analysis of the uric acid is the example of these amperometric sensors. The formula of urea is observed here. NH2 single bond C double bond O single bond NH2 is the formula of the urea. Urea plus or uric acid plus H2O plus oxygen undergoes reaction with urease to form allantoin H2O2 plus CO2. H2O2 means hydrogen peroxide. Again this hydrogen peroxide undergoes oxidation to form 2H plus O2 plus 2 electrons. Applications of these sensors. We use these sensors in the food sample analysis, parametrical analysis to identify toxic chemical substances like methyl isocyanide and also carbon monoxide, which are toxic chemical substances. Analysis of the biological sample. First one is glucose, urea, and the blood sample, etc. are analyzed by these sensors. Identify impurities present in the water, salts, etc. Here in this diagram there is a sensor which is connected to this human body, and with this sensor we are going to receive the here this is the sensor connected to the body, and through this sensor we are going to detect the glucose present in our body in the glucometer device. Advantages of these sensors. No tools are used to convert chemical information into the electrical signal. There are no tools. We use no tools to convert the chemical information to electrical signal because we use sensors. These sensors are high sensitivity and high selectivity and high surface area. Here we see in the diagram there are high sensitivity, high selectivity and high surface area. When these sensors are used, no tools are required to convert chemical information into electrical signal. These are important to our daily life. Thank you for giving this golden opportunity. Thank you.